Hello, everyone. My name's Phil Bourne. I'm actually a professor in the Skaggs School of Pharmacy. I do early stage drug discovery. I'm also the Associate Vice Chancellor for Innovation and Industrial Alliances here on campus. And that's a new role for me, something since this class was given last year. And I have quite a different perspective on things than I had before as a result of that. And I'm not going to touch on too much of that today, but it's clearly something that is nagging at me right now in terms of how we get things out of the university into clinical trials and, of course, beyond. I think there's some huge challenges there, and I've heard a bit about, you touched on that already, and perhaps we could talk about that at the end. Okay, so I'm going to focus on the idea of proteomics and genomics and what that brings and how that's perturbing the drug discovery process. And I would say that you've kind of heard a bit of this. Let me just cast it in my own way by saying it's the best of times, and in some ways it's the worst of times. And I love this. This is the opening paragraph from Charles Dickens' book, and it's so apropos for drug discovery. It's unbelievable. It was the best of times, it was the worst of times, it was the age of wisdom. And I think genomics and proteomics is kind of giving us the age of wisdom, but there's also the age of foolishness in how we've so far been able to take a lot of that forward, because we haven't changed the way we do things in large part in the drug discovery process, even in light of this new data and these new capabilities, in my opinion. And I'll give you some illustrations of why I think that's breaking down. And I could go on to this with the winter of despair and so on. But here's the sort of best of times, right? We are in this omics revolution. It started in about 1990 with the Human Genome Project, which took quite a long time to actually bear fruit after it was finished in 2000. But there's no question, and there's no doubt, and you asked the question about personalized medicine, that as we move down, we're at whatever it is now, $3,000 to $5,000 for a genome, human genome. We will be down at the $50 level in the next several years. That is going to have undoubtedly a profound impact on healthcare in times to come. So from that point of view, it's the best of times. And we'll see what that means in terms of data and integration, the kinds of other questions you were asking. In some ways, it's sort of the best of times because, we have seen through NIH and other places and the private sector large increases in research budgets, in principle spurned by the human genome and other things, to actually do better at discovering drugs. But then, as you've already heard, I'm sure, and you'll continue to hear throughout this, the success rate is abysmal. And, so this is another sort of illustration of the best of times and the worst of times. So putting that into words, we have this explosion of data, which in principle allows us to do a lot. We are starting to get, and I'm going to emphasize this, we're starting to get to an understanding of complex systems in ways that we never have before. That should be impacting the drug discovery process. At this point in time, I don't think it is, but I think we're on the cusp of a turnover, and I'll say more about that. And of course, the information technology that's needed to do all this, which is an area where I spend quite a lot of time myself, is obviously improving. So let's optimism rule. So what this all means is, this whole traditional model that's been based on, in the computational side at least, computational chemistry and cheminformatics, we're now bringing the idea of bioinformatics and systems biology, studying the whole system, particularly in silico, into play into this whole process. And the end result of that is something I would call systems pharmacology. and it's actually doing pharmacology in a different way than we've done before, which I'll illustrate. And I'll show you a couple of examples of what I mean by that and why I think it's so important. All right, so what's my agenda? I'll give you a little sense of, I think what you're going to get out of this course is, because this is the nature of the game right now, you're going to get, from different people, you're going to get some mixed messages. Their opinions are going to be somewhat different about where drug discovery and the whole industry is going. So I'm just giving you my perspective. I'm going to tell you where it comes from, and you can take it or leave it. That's the business. I'll say a little about the Omics Revolution in a bit more detail, something that I'm very passionate about, and something that I think is also driving some change. And anything that drives change, I love. So the notion of open science, I mentioned the IT revolution, but open science is also something that's changing. Drug companies are becoming, at some levels, much more open than they ever were before. And that's basically out of necessity because of failure, my opinion. Again, this is bias coming in, right? And then what impact does all that have on drug discovery? We'll look at that, and I'll give you a couple of examples, because how could I possibly stand here and not talk about my own work? People who work, of course, I don't actually do anything, but the work of people in the lab. And then there's a few words of caution about everything I've said, which probably means it's not true at all. All right, so what we do is I maintain a resource, an open resource called the Protein Data Bank, which is the repository for all macromolecular structure information, proteins, DNA, RNA. And there hasn't been a drug discovered in the last 30 years that hasn't used that quite extensively. And that's given me a certain viewpoint in all of this. We distribute a ton of data. There are 7 structures looked at every second, essentially, around the world. And then we've been using that ourselves in a research perspective to do what I've already introduced to you, the notion of systems pharmacology. So, and personally, I mentioned that open science. Also, I've started some companies while I've been here, which is one of the reasons why I'm doing this Associate Vice Chancellor job right now, because you'll learn as you go on, the more you complain about something, if you complain long enough, before you know it, you're part of the solution. So trying to form companies here is not totally straightforward. So finally the Chancellor says, well, you don't like it, fix it. So that's kind of what I'm trying to do right now. So that's sort of some background. So let's look at the omics driver. So right now, we're in a very interesting situation associated with data, particularly genomic data, but it's true of all data in biology and beyond, for that matter. We're completely out of whack with the ability to handle that data. So the data, you all know what Moore's law is? Yeah, most of you. So Moore's law is just a reflection of the dropping cost per some unit of computing over time. And essentially, what you could do with a unit of computing, whether it be storage or CPU, was essentially that doubles every 18 months. And that it's gone between 12 and 24 months, but 18 is a sort of average. And biological data of all ilks, and the whole notion of translation and having data not just at the omics level but at higher levels, all of that data for a long time that was essentially keeping track with paralleling Moore's law. So the cost of maintaining that data was essentially on par, hadn't changed over time. That is no longer true. In fact, with 4th generation sequencing, the newer types of sequencing technologies, the amount of data far, far outweighs. This is an exponential scale, and you can see, sequencing sort of was paralleling this for quite a while. And then, first of all, with 2nd and 3rd and 4th generation, that's really gone completely wacky. Right now, the amount of sequence data that we're generating is doubling every 5 months. It's absolutely huge. And that doesn't speak to any of the datas of greater complexity, which I'll get to in a minute. No one is really addressing the issues of what we keep, what we throw away. We're not addressing issues associated with the complexity of that data, the errors in that data, how we represent metadata, how we represent provenance, which is how we describe the value of that data and who owns it. All of these things are completely out of whack right now. Not only are we generating large amounts of data, but we're generating data of much greater complexity. So this is this protein data bank I mentioned, and this is its growth in terms of number of structures that are in it. Also, well, not every one of these, but the majority of these little blobs are actually Nobel Prizes. So the myoglobin from John Kendrew won the first Nobel Prize in structural biology. And all of these essentially, most of them also brought forth Nobel Prizes. These are actually drawn to scale. So you can see what's happened over time is the complexity of data up to the whole ribosome, for example, the whole protein synthesis factory in the cell. We now understand that in ways that we just never did before. And so the complexity of this data and what that means in interpretation and use, which I'm not really going to get into, but except to say we are in an era that's quite different than it was before. I've just had a discussion with the Chancellor last week about actually establishing a data science department here, or some entity that's dedicated to data science. And people would say, why do we need that? Don't people just do that in their labs? Well, I think we need it. You could have said the same about computer science 30 years ago. Computer science became a very vibrant and active discipline. And I think what you're going to see is data science become the same kind of thing, because it's actually critical to the future of many industries, including the drug discovery industry. Again, my biased opinions. A number of years ago, I was actually asked by the then Chancellor to tell me what bioinformatics is in one slide. So I produced this, and it never got used because it's way too complicated and way too, there's not a picture on it or anything like that. So, but it really sort of reflects things that are going on even today. So I already mentioned this notion of huge influx of data. And what we're trying to do, of course, is turn that data into information, knowledge and discovery, including in drug discovery. And, these are sort of the processes that go on to reach that. At the same time, back in, it was about 2010, we actually surpassed the number of websites. There was more websites than there were people on the planet. So, we've got this incredible increase of amount of information available. And at the same time, we're actually working at higher levels of complexity to understand what's going on, all the way to higher life forms, all the way between that and DNA sequence. And all of these projects in here, brain mapping, cardiac modelling, are things that are going on this campus that really relates to that kind of integration of data. So what you'll see coming out of all of this as I move forward is, first of all, I believe it's changing the way that we do science and the way we think about science. We've been siloed for way too long. Each of these different levels of biological complexity. What we're starting to do now is move across those levels of complexity. And, it's a very exciting time. It is the best of times, at least for that. So this is just, I just love this. This is one of my, this is my dream of doing science. So Craig Venter, you probably know, has the Venter Institute here. He was one of the major proponents of metagenomics. And essentially one day he just decided, I'm going to take my yacht and I'm going to sail around the world, and periodically at all these dots I'll scoop up a bucket of seawater. And I'm going to sequence everything that's prokaryotic, everything that's small in that seawater. And so what he did, and then of course he comes back, so he goes on this great cruise on his own yacht, and when he comes back he's got a science paper already. It just doesn't get any better than that. In any case, so this is where he went, and he collected all this data, and so we suddenly started to accumulate very large amounts of protein sequence data that we didn't have before, because we were sequencing these complete organisms. And so there was of the order of 17 million sequences on the first pass that we'd never ever seen before. Right? So there was 17 million completely new proteins that no one had ever seen before. And 99% of the DNA associated with that was from unknown organisms. We had no idea what they were. So we hadn't even begun to catalogue one tiny fraction of the organisms that are in the ocean. So the idea that there is culturable organisms there, that there are compounds that can be found, new receptors, and all the kinds of impact that it could have on drug discovery is all there. All right? But as we started looking into this, there were a number of discoveries, which I won't get into. But what it did is it started the whole metagenomics revolution. So it wasn't just getting buckets of seawater. Everybody wanted to have their own yacht, right? So they basically started scooping up and doing things. They started doing soil, right? So they were analysing all the organisms in a cubic foot of soil in different parts of the world. And perhaps most interesting, of course, was actually to look at the human gut. So the microbiome project is actually sequencing all of the organisms that exist within the human gut. Already out of that, we have some pretty good biomarkers for certain types of autoimmune diseases like Crohn's disease. So you can actually now, we're getting to the point of early detection of Crohn's by virtue of doing large-scale sequencing of the human gut microbiome. So, I think this is, exciting examples of what's to come. This just illustrates the known and unknown. So this is just one particular family of proteins. I don't want to get into a lot of detail, but you can see that the red were completely unknown. This is a phylogenetic tree, so it expresses the evolutionary distance between these things. But the only point is that higher eukaryotes weren't sequenced, but in this metagenomic data, we discovered thousands and thousands of these new PTPases. That was just one protein family. So lots of very valuable and new information. But with all of that comes problems. You know, it's always the case. When things move very quickly, it takes a lot of time to catch up. I mean, it takes times to catch up ethically, legislatively, and, you know, in terms of technology. So as a result of all of this, for example, when you go to a database and you pull out a sequence and you look at its annotation, there are estimates to say that 30% of that annotation is wrong, because that annotation has been made by propagation. So in other words, if someone says, okay, this particular protein sequence does this, and then someone else compares it to another sequence and they say, okay, it's the same, this must do the same thing. If the first one was wrong, the second one's wrong, and you get this kind of propagation. So, you know, we've got a lot of misinformation out there right now. That's the other point. So in any of these studies, it's very important to take heed of all of that. So that's sort of the omics revolution part of it. Let's look a little at some of the other revolutions that are going on, which I think are going to impact things. So the open science and the IT revolutions, what does that bring to the table? So who's familiar? I can ask a question, but you're not allowed to answer. Only put up your hands. Who's familiar with open access and what it means? Okay, a few people. How many people have published a paper in an open access journal? Well, you should. So the basic idea is we're changing the publishing paradigm. We're making information accessible to absolutely everybody. And so instead of paying to read it, you're paying to write it and publish it, essentially. The models that do that, the journal models and the societal models, all still work. There's now proven business models to support this. But it changes what you can do because, for example, a typical license on an open access journal article would be what's called CC BY. It's a Creative Commons license, and these are actually declared and described accurately. And it means that anyone can take your knowledge and they can do what they like with it as long as they attribute you as the original source of that information. So the ability to propagate knowledge actually, in my opinion, increases quite significantly. And, you know, it's taken a while for it to get going, but last year about 20% of all journal publications in the biomedical sciences were open access. And that number is increasing steadily. I think in 5 years, essentially, open access will be the model. And so that creates a lot more access to information than we ever had before. I think there's some interesting software is sort of going along the same paradigm. I won't get into that. How many people are familiar with GitHub? That's a good reason. One person stretching their fingers. Okay, so I won't get into that. But I mean, other areas which I think are very intriguing are the socialization of science and also the flattening of the hierarchy, such that anyone could make discoveries and be listened to. Right? I love this fact we're in this generation. You know, when I was growing up, I'd be sitting at the dinner table and my father would slap me around the head and say, "You should be seen and not heard." Right? Now, of course, you know, I couldn't possibly do that with my kids. Well, they're too big now anyway, but when they were smaller. But no, we went, long time ago, we went into the generation of not being seen and not heard, but now we're in the generation of heard and not seen. I mean, I've got to tell you this because to me it's really interesting. I got a paper into the journal that I run, and it was a fabulous paper on pandemic modeling. All right? And I was so impressed by it when I looked at it, is I arranged to meet the author because it was a single author, and she was actually here in San Diego. We met, we talked about the paper. I was so impressed with her, I invited her. She gave a lecture here at UCSD. She went to Princeton to give a lecture on the same stuff. Really nice. So what? What's special? She was a senior at La Jolla High School. She was 15 years old. That paper was reviewed by Science. So anyone can do great stuff now. They have access to information, they have access to knowledge, and under certain circumstances, obviously she's an exception rather than the rule, but it tells you the kinds of things that are coming. And so, in my opinion. So all of this is perturbing the system. And we're starting, so what does that all have to do with drug discovery? Well, there are a series of initiatives. How many people here have heard of Sage Bionetworks? Okay, so this kind of brings together the number of the things that I've been talking about. You know, the use of the commons, which puts into the public domain lots of biological data, genomic data, patient data, and everything in between. There is an open framework, there are open licensing to make all of that accessible. And so this is disease models that are going online and being accessible. And so the idea, it's founded by Stephen Friend, who actually founded Rosetta Pharmaceuticals, which was a spin-off from Merck. So, you know, it's someone who's been along the traditional route and now is dedicated to the idea of creating open drug discovery capabilities. And I think that's kind of an example of bringing that together. When I gave this lecture last year, we were actually just in the point when, if you remember, Wikipedia was closed down for a while, and there was a huge ruckus, and I think that's just another sign that the open access battle was being fought, and it's not been won yet, but it's kind of looking good from my point of view. All right, so what's all this? We've sort of seen what's going on. We've seen the notion of a Sage Bionetworks as an open consortium for drug discovery. How could this potentially impact drug discovery more directly? So I don't want to necessarily answer that question, but I want to go back and I want to look at the question is why is the drug discovery process, why are we not discovering many new drugs? There are economic reasons. I want you to tell me, but you can't tell me. But you could tell me, but you can't tell me for whatever reason. But here's a couple of data points that got to get you to start thinking. All right, gene knockouts. When you knock out a gene, only in about 10% of cases do you actually see an effect on the phenotype. Why is that? Because there's redundant functions. There's alternative network routes. There's robustness in the interaction networks. So that's already got to ask you, so okay, you're knocking out a gene. What if you in some way perturb the system with a drug so you hit a particular target? Well, in many cases, because for these exact same reasons, you might be having very little effect. You know, you can't ignore these things, all right? And then when you look at the fact that if you take all of the biologically active compounds and you start looking at them and you assay them in various ways across panels, you find just from that group, 35% of them bind to more than one target. So this whole idea of the premise of the drug discovery paradigm for so long with respect to small molecules of one drug, one target, one disease, this kind of data just says, how can that possibly work? To me, it explains exactly why when I turn on the TV and try and watch the news, most of the time I'm hearing about the side effects of drugs that are being advertised during the commercials. And the reason there are these side effects, in large part, has to do with these kinds of effects. All right, so what should we do about it? What has the drug industry done, in my opinion, for quite a long time? It tried to ignore it. It kept trying to chase after these blockbusters that were these, and there are a few out there. Many of them being discovered serendipitously after all of this massive amounts of research, but they're all out there. And a lot of them are actually natural products. They're not actually synthesized drugs. So, but if we accept this notion that there isn't one drug, one target, one receptor, what can we do? So here's something that actually drives this home even further. All right? This is the human genome tree. So you and I have about 550 human protein. We're humans, so we have 550 protein kinases approximately. This is the phylogenetic tree of those kinases. Each one of these instances and the balls on it are kinase inhibiting drugs. They're on the market, FDA-approved drugs. And so you can see, Gleevec, which is a wonder drug, and the size of the ball here is a reflection of the binding affinity to a target. A number of these drugs actually bind to off targets with higher affinity than what the known target was believed to be. And then you take something like staurosporine, which is a natural product, that binds to huge parts of the tree. So what does that mean? It means that you're not inhibiting a single target. What you're seeing is a massive collective effect. So then the question becomes, well, how can we use that? If we accept that notion, what can we do with it? So this Ehrlich philosophy of the magic bullet I think is not being realized. So it's just naive in a complex system, in my opinion. All right, so how can we apply that? How can we accept that? And then what can we do about it? So I'm going to give you a couple of examples from our own work. All right? So if we accept the fact that we've got this multiple binding, let's try and figure it out, what it is. Let's say I've got a drug, it's this right fist, I've got a receptor, and these things are binding together. I know what that receptor looks like now. I know what its binding pocket looks like because I have the complex. So let me take that binding site and let me search across the complete structural proteome, the proteome of the human, looking for all instances of a similar binding site. If I find that, then it tells me one of two things, three things. It may tell me nothing, but it might help explain the side effect of a drug because I now know another target. But even better, from a drug discovery point of view, it might actually allow me to repurpose that drug because I've discovered a target that's actually associated with a different condition. And I have examples of all of those. I'm going to try and tell you a little bit about a couple of them. So these are the kinds of things it tells you, and I pretty much described that. So the basic idea is in this database I mentioned, there are things like Lipitor, $100 billion a year drug. It's sitting in a public database. And so you can go, in this PDB I mentioned before, so you can go and look at it bound to its target, and you can play around with that. So you can take that target and you can look for other similar targets. And so, there it is. And so what you do is you characterize the binding sites, and then you essentially search for those binding sites across all these other proteins, and you do the statistical test to see how valid what you discover is. How valid is this alternative binding site statistically? I'm not going to go into any of the science of this. This is work that's been done by Leiji in the lab. I just want to say that. And, so that's sort of the technology. I'm not going to go into any of the details. So what can you do with it? What are the kinds of things you can do with that technology? So we happen to be interested in neglected tropical diseases, partly because, of course, the reason they're neglected is hardly anyone is really looking at them. The drug companies are not, until recently, haven't been looking at them very much because there's no money in it. And yet, on the other hand, there's 1.7 million deaths a year approximately from tuberculosis. We also, people worry about it constantly because we have multiple drug resistant and we have extreme drug resistant strains that, could eventually get into this is a developing world disease, and unfortunately it doesn't get the attention it should. But if it gets into the developed world and these strains start really proliferating, we've got a lot of problems because there hasn't been, until recently, particularly successful new treatments for TB for 40 years. So this just happens to be something that we would focus So this is just an example of this kind of large-scale biology and what you can do with it. And then I'm going to tell you some of the limitations of it. All right? So what you can do is you can say, all right, I'm going to take, we have the genome of various strains of TB. From that, we have the protein coding regions. We know what the proteins are. We have only 200